Hi guys, welcome to Mastermed Academy. If you are new to my channel, please hit the subscribe button and press the bell icon to receive future updates on our new videos. This is the part 2 of adaptive immunity. If you haven't seen part 1, I would suggest you guys to go have a look and understand the first part of this lecture. So guys, today I'll be talking about T cells. How does a T cell in the thymus maturates? And what are the steps? What is positive selection? What is negative selection of a T cell in the thymus? I'll be also talking about the different type of T cells that we should be aware of. What are the different T cell receptors? And finally, I'll be talking about T cell activation. What are the exact signals that are required for a T cell to get activated? So let's start. So guys, let's start with adaptive immunity part 2. Now, this is a beautiful picture of a T lymphocyte here. I thought I should put a picture of a T lymphocyte exactly how it looks. So this is how a T lymphocyte looks like. Now, what are T cells? So T cells recognize peptide antigens that are displayed by the MHC molecules. Now, peptide stands for protein antigens. I previously mentioned in part 1 adaptive immunity that in order for a T cell to recognize an antigen, it has to be a protein antigen. It has to be a peptide antigen. It can't be a non-peptide antigen, right? So bone marrow is the site of progenitor stem cell development for both T and B cells. I like to think of this like a site where baby cells are formed. So baby T cells and baby B cells are formed in bone marrow. Now if the T cells wants to further differentiate into a specific type of cell, that process happens in a place called thymus. So thymus is the site for progenitor T cell to differentiate into a mature T cell. So T cell differentiation takes place in the thymus and T cell development takes place in the bone marrow. Now T cells belong to cell mediated immunity, while the B cells belong to humoral immunity. So I hope you guys remember that. T cells are responsible for mediating type 4 cell-mediated hypersensitive reaction. This is a beautiful picture of a T cell, 3D picture, like I showed a neutrophil, if you remember, in my innate immunity. So you can take a moment to appreciate the picture of the T cell. Now this is a picture where you can see an RBC here on the left side. The middle yellow part is a platelet, and the right part of the picture, the blue cell, this is another T lymphocyte. It is quite similar to the picture I showed you in the first slide. So this is also a T lymphocyte. So let me write it as T cell. Now T cell maturation occurs in thymus. T cells begin as hematopoietic precursor cells in bone marrow. Again, the baby T cells are found in bone marrow, and after development they migrate to the thymus as thymocytes. Now why you want to go to the thymus? Because you want to get differentiated into a specific type of T cell. In the thymus, they undergo a process of selection which determines their fate. Now what is this selection? Let me explain you guys. Now this is a beautiful cartoon here that explains the T cell development and differentiation, basically more of differentiation, right? Now let's imagine that the thymus is an exam center. So let me write exam center here, and thymus is the exam center. Now the thymus has two regions. It has a cortex and it has a medulla, and in the cortex there is a process called positive selection. So exam number 1 that the T cell has to give in a thymus takes place in cortex, and the name of that exam is called positive selection. The second exam the second exam the T cell takes is in the medulla, and that is called the negative selection. So the first exam is named as positive selection and the second is named as the negative selection. Now when the T cell precursor or the baby T cell develops in the bone marrow, through the blood circulation it goes to the thymus. Now in the thymus you have the cortical region here as shown in the picture. Here is the exam center number 1, and the T cell is undergoing a exam called positive selection. Now, what is a positive selection? Now, in positive selection, we have a T cell that is double positive. Now, what do I mean by double positive? Double positive means that the T cell has both CD8 positive and CD4 positive characteristics, as shown in this cartoon here. Now, positive selection basically means that the T cell that expresses the T receptor or the TCR, the T cell receptor, it binds self-MHC molecule. So let me draw a picture here. So let's imagine that this is a T cell here and it is expressing a TCR or a T cell receptor. In positive selection, this T cell has to bind basically a self-MHC molecule. So those cells that bind the self-MHC molecule, they will survive. And those cells that don't bind self-MHC molecule will undergo apoptosis. So I hope the positive selection is clear. Now, let's assume that the first the cell here in the cartoon, this passed the exam of positive selection. The second exam name is called the negative selection. Now, in the medullary part of the thymus, the T cell being either CD4 positive or CD8 positive, they undergo a process of negative selection. Now, in plain words, negative selection means that the T cell expresses its receptor TCR. Now, if a T cell expresses high affinity for self-antigen, that T cell will undergo a process called apoptosis. Now, this is negative selection. So, let's imagine that this is another T cell and here is the TCR, and let's imagine that this is a self-antigen. So, if this T cell binds this self-antigen, this will undergo apoptosis. So, the cells that don't bind the self-antigen, they pass this test and then they go to the lymph node through the blood circulation. So, once they reach the lymph node, the T cell will actually differentiate into either a T helper cell or it can differentiate into a cytotoxic T cell. So I hope you guys understood the mechanism of positive selection, negative selection, how exactly it is carried out in the thymus and why it's important. So you can go back in the video for 4 or 5 minutes and you can see again the whole process if you didn't understand. So let's go to the next slide. Now this is again the same table and it says whatever I just said in the previous slide. So in the positive selection, the process takes place in the cortex. Negative selection, the process takes place in the medulla. The T cells expressing TCRs, which can bind self-MHC, will survive. This was what the positive selection was. And the negative selection is basically T cells expressing TCRs with high affinity for self-antigens undergo apoptosis. Remember, apoptosis is a programmed cell death. It's not like necrosis, because necrosis is basically due to trauma or injury to our body and it's not mediated by our own body. Whereas apoptosis is mediated by our own cells, our own body. So this is called the programmed cell death. Some people call it apoptosis, some people say P is silent, so they call it apoptosis. However you want to call it, it's up to you. So the self-antigens which were expressed in the process of negative selection, they are expressed due to a gene, and the name of that gene is called autoimmune regulator AIR gene, AIRE. So AIR gene is responsible for expressing self-antigens in the thymus, especially in the medullary part of the thymus, that expresses the self-antigens basically, right? So what if the AIR gene is deficient? What will happen? So when you have an AIR gene deficiency, we have a disease called autoimmune polyendocrine syndrome type 1. So let's see what that is. Here is the autoimmune polyendocrine syndrome type 1. I hope you now know the spellings of it. Autoimmune disease due to deficiency of AIR gene. So there is a deficiency of the AIR gene, as I mentioned. Now this is an autosomal recessive disease and it is present on chromosome number 21. Now what are the classic hallmarks? You have to remember this for any exam that you have regarding immunology. The classic hallmarks for autoimmune endocrine syndrome type 1 are, first is chronic mucocutaneous candidiasis. Candidiasis or candida, you know that this is a fungi. So of course you give antifungal to kill this, right? Mucocutaneous candidiasis, then this is chronic. Second is the hypoparathyroidism, and this is a state where you have decreased parathyroid hormone, and that leads to decreased calcium in your body. The third is the adrenal insufficiency. This is a process that affects the adrenal glands, and that is unable to secrete certain types of hormones, let's say. So I hope you understood the hallmarks of APS type 1. You can call it APS type 1. Now, what are the types of T cells? First, we have a helper T cell. This expresses CD4 receptor that will recognize the antigen expressed on MHC class 2 molecule, which I already told you. So if you have a cytotoxic cell, it will express CD8 receptor and recognize the antigen displayed on MHC class 1 molecule. So remember that cytotoxic T cells will recognize antigens displayed by MHC1, not MHC2. So remember, it's MHC1. Regulatory T cells express CD4. They have high affinity for interleukin 2 receptor, and they recognize antigen displayed on MHC class 2. So I hope you guys understood this slide. Now this is another cartoon showing that CD4 T cell binds antigen which is presented by MHC class 2, and that will lead to the formation of mature helper T cell. On the other hand, if you have an immature CD8 T cell, that will bind the antigen presented by the MHC class 1, and that leads to formation of cytotoxic T cells. So I hope you guys understood this cartoon. It's very simple, trust me. So what is a T cell receptor? Now, how does it look? So a T cell receptor consists of alpha and beta chains, 2 alpha and 2 beta chains. It also has a CD3 complex, right? Now, T cell receptor are encoded by genes that rearrange for diversity. This is a process called VDJ recombination. If you guys haven't seen my previous video, part 1, regarding adaptive immune system, I will recommend you guys to see it, because I explained exactly what is this VDJ recombination. But just to give you a short version of that, VDJ recombination is a process where we try to make diverse receptors, i.e. different kinds of receptors, to bind to different kind of antigens that are present in the world. Okay, so we require this VDJ recombination or VDJ process where we can shuffle genes and make new kind of receptors for different type of antigens out there. So VDJ recombination affects not only T cells, but it also helps to form antibodies, i.e. immunoglobulins. So different types of immunoglobulins with different type of FAB region. Now, just to summarize, what is an FAB region? This is the heavy chain, this is the light chain of an antibody, this is the FC portion of the antibody, and this is the FAB region of the antibody. So also the VDJ recombination helps the antibody to form different kinds of FAB region, right? So that we can bind different types of antigens which are present out there in the world. So I hope you understood the VDJ recombination in a nutshell. V stands for variable region, D for diversity, J for joining, and C for constant. Now, this is a structure of a T cell receptor. As I mentioned, there are 2 alpha and 2 beta chains. So here we have 2 alpha chains and we have 2 beta chains here, along with CD3 receptors on the side. Now, what are the subsets of T cells or different subgroups of T cell, right? So the first one is a T helper 1 cell. T helper 1 cell activates macrophages, very important to know. They also activate the cytotoxic T cells and NK cells. I would recommend you guys to remember the exact cytokine secreted by the T helper 1 cell, which is interferon gamma and interleukin 2. Now let me draw a diagram to make you understand what is the relationship which is shared between a macrophage and a T helper 1 cell. So let's imagine that this is a macrophage. I know the drawing is ugly, but still. And let's imagine that this is a T cell, right? Especially T helper 1. Now T helper 1 secretes interferon gamma. Now you have to remember that interferon gamma is responsible for activation of a macrophage. While once a macrophage is activated, the macrophage will release another cytokine called interleukin 12. interleukin 12, and interleukin 12 helps to induce and activate T helper 1 cell, right? So you have to remember this intimate relationship between T helper 1 and a macrophage, because there are certain diseases where we have a decreased amount of interferon gamma or we have a decreased amount of interleukin 12. So remember this intimate relationship. Now, T helper 1 cells are induced by interferon gamma, as I mentioned, and a interleukin 12 here, right? Now, it is inhibited by interleukin 4 and interleukin 10. Now, this T helper 1 is important for intracellular organisms like Mycobacterium tuberculosis and Listeria. Now, why is that? Because imagine if there is a situation where we have a T helper 1 cell that is unable to secrete interferon gamma in sufficient amounts. So, do you think that if there is less amount of interferon gamma, will the macrophage be able to activate itself? No, because you don't have enough of interferon gamma. So if a macrophage is not able to activate itself, do you think it is going to be able to eat the pathogen, cause phagocytosis, or like kill the bacteria? No, it maybe in small numbers some macrophages might do it, but largely it's not possible because there is not enough interferon gamma to basically activate the macrophage. So granulomatous diseases can occur as macrophages secrete tumor necrotic factor alpha, which promotes granulomas. Now, in one word, or let's say in one sentence, I can say tumor necrotic factor alpha is a cytokine that is secreted by macrophages that promotes formation of granulomas, and it also promotes inflammation. So you have to remember that. Now, interleukin 12 deficiency, this is one type of deficiency that I mentioned, and this is the second type of deficiency that I mentioned in the previous slide. So interleukin 12 deficiency, we have a loss of activated T helper 1 cell to produce interferon gamma. We have a very weak T helper 1 response and low interferon gamma, and this leads to susceptibility to disseminated mycobacterium infections, and BCG vaccine is increased. So it is not really appropriate to give a BCG vaccine to a patient who is interleukin 12 deficient. Now, what is the treatment here? It's very simple. You give interferon gamma that will lead to the activation of macrophages. So you give interferon gamma. So in my opinion, interleukin 12 deficiency is a lighter version, or it's like a non-harmful, like a less harmful version as compared to interferon gamma deficiency. Now, if you have an interferon gamma receptor deficiency, then you have a severe disseminated mycobacterium infection. Here is not the susceptibility. Here you will for sure have severe dissemination of mycobacterium. So that's why the treatment here is the stem cell transplant. So the second type, the interferon gamma receptor deficiency, this is more dangerous than interleukin 12 deficiency. Okay, so I hope you guys understood that. If you don't understand something, or if you have questions, please drop your comments below and I'll try to answer them as ASAP. Yeah, now the second is a T helper 2 cell. Now T helper 2 cell produce IgE and they help us defense against parasites and they activate the eosinophils. Now T helper 2 cells secrete interferon 4, interferon 6, 5, 10 and 13. And if you guys remember that T helper 1 is inhibited by interleukin 4 and interleukin 10. So let me go back 2 slides and show you guys here, exactly here, inhibition by interleukin 4 and interleukin 10. T helper 2 are induced by interleukin 2 and interleukin 4. Interleukin 2 is formed, or let's say secreted by T helper 1 cells, inhibited by interferon gamma, T helper 2. Okay. Now the third type of cell is the T helper 17 cell. This induces neutrophilic inflammation and this provides defense against extracellular organisms. This secretes interleukin 17, 21 and 22. T helper 17 is induced by TGF beta, interleukin 1 and interleukin 6, while it is inhibited by interferon gamma and interleukin 4. Now you have to remember for your exams that if you have a deficiency of T helper 17, the deficiency will lead to hyper IgE syndrome or a job syndrome. This is basically an immunodeficiency syndrome, which I'll be talking about in a separate module. I have made a module separately for immunodeficiencies, so we'll take this case there. So don't worry about that. The fourth is the T regulatory cell. Now, T regulatory cell helps to maintain tolerance to self antigen and prevents autoimmunity. So T regulatory, is primarily responsible to prevent autoimmunity. It secretes TGF beta and interleukin 10, induced by TGF beta, interleukin 2, inhibited by interleukin 6. This suppresses CD4 and CD8 T cell functions. So if you obviously suppress the major players like CD4 and CD8 cells, there is of course no immunity, so it prevents autoimmunity, right? No immune function. It's identified by expression of CD3, CD4, CD25 and FOXP3. I would like you guys to remember FOXP3 because there is a disease regarding this FOXP3. So let's see what it is. IPEX syndrome. IPEX stands for immune dysregulation, P stands for polyendocrinopathy, and E stands for enteropathy. X is X-linked. Now this is due to a deficiency of FOXP3 gene. So when you have a deficiency of a FOXP3 gene, it leads to autoimmunity. So this is an autoimmune disease. Now this is associated with diabetes in male infants. Very important. So this is characterized by nail dystrophy, dermatitis, autoimmune dermatologic conditions, enteropathy and endocrinopathy. Now dermatitis or the skin problem could be anything, but most commonly we see eczema. Now here is a picture of an eczema here. Let me write eczema. This is the spelling. So this is a type of a dermatological condition seen in IPEX syndrome. The next is the cytotoxic T cell. It is CD8 positive cell that binds MHC1. I hope now you have already memorized since I've said it so many times. Cytotoxic T cells kills neoplastic cells, donor graft cells by inducing apoptosis. Now this fast ligand pathway is one of the few processes that is involved in the apoptosis of a particular cell. We have many types of processes. There is like caspases, then this is a fast ligand pathway, and there are many things, but we'll discuss it in some other module. But for now, trust me that the cytotoxic T cells use the fast ligand pathway to induce apoptosis. The T cytotoxic cells release granules containing perforins and gran enzyme B, right? And they obviously help the T cell to basically kill the pathogen. So the next slide here explains us the cell surface proteins, the T cell and the surface proteins here. Now the T helper cell basically has CD4, CD40 ligand and CXCR4 oblique CCR5 as the surface protein. T cytotoxic cells have CD8 surface protein that binds the MHC1. The T regulatory cells basically have CD4 and CD25. They are the classic markers for T regulatory cell. If you really want to remember just one classical marker for T regulatory cell, it has to be CD25. You have to remember that. So T cell activation, this is the last couple of slides and we're done. The T cell activation has APC. APC stands for antigen presenting cells, and they could be B cells, dendritic cells, macrophages, Langerhans cells. Two signals are required to activate T cells. I like to think of it as a signal number 1 and signal number 2. Signal number 1 is the primary signal or like the main signal. Signal number 2 is a secondary signal or a co-stimulatory signal. If you just have signal number 1 but no signal number 2, you still won't be able to produce the cytokines. So let's see, what is this signal number 1? What is this signal number 2? So let's have a look. Signal number 1 is a T cell activation. So let's imagine in this slide, as you can see, we have a dendritic cell. There was this antigen, and this antigen went into the dendritic cell, got processed and expressed in MHC, either class 1 or class 2, we don't know, right? So let's imagine that this is an MHC molecule. Now in signal number 1, there is an interaction between the MHC molecule and the T cell receptor that recognizes the antigen presented by the MHC molecule. So the area that I marked, this is signal number 1. So this is signal number 1. And the second signal, which is the co-stimulatory signal, is responsible for production of cytokines, and they are mediated by the CD80-86 receptor on a dendritic cell. and CD28 receptor on a naive T cell. So this is signal number 2, or you can also call it co-stimulatory signal. You need a co-stimulatory signal to stimulate the process of to stimulate production of cytokines. So let's go back. Signal number 1 is a T cell activation signal. APCs with their process antigens drain to lymph nodes. Antigen will be presented exogenously on MHC2 and is recognized by CD4 cells. Endogenous antigen is presented on MHC1 and it is recognized by a CD8 T cell. If you remember a picture that I presented in the last video regarding part 1 of adaptive immunity, I mentioned this endogenous antigen processing and how it happens and eventually how it is expressed by the MHC class 1. You can refer to that video and see. Signal number 2 is the proliferation signal, also called the co-stimulatory signal. This involves the interaction of the B7 receptor on APC and CD28 on naive T cell that leads to stimulation of cytokine production. So again, this is the same picture, and I hope you understood exactly how the T cell is activated and what are those two signals and what are the respective receptors that are responsible and takes part in this activation of T cell. So I hope you guys enjoyed this video as much as I did, and if you have any questions, if you did not understand something, please drop your comments and I'll try to reply to you guys as fast as I can. And let me know your thoughts, and thank you for your attention. And please do not forget to click the subscribe button, press the bell icon, and I hope to see you guys with the next video on Mastermed Academy. Thank you so much. Bye bye.